Okay, I'd like to welcome everybody here. It's straight up noon. This is our monthly Dairy Team webinar. Today's program will focus on feeding red seaweed to mitigate methane production in the dairy cow. Today's presenter is Dr. Ranga. He's assistant professor at Iowa State University. He's got an animal science and dairy science background and is recipient of the 2023 Early Achievement Teaching Award from the College of Ag. He teaches a large undergraduate animal nutrition class, advises undergraduate students, and is involved with training undergraduates in research under the Honors in Science of Practice programs. Dr. Ranga will discuss feeding red seaweed and how it's been shown to decrease methane production. He's also going to talk about strategies for commercial farm and the multiple challenges to mass production, palatability, and milk safety. As I always do, I'll say, Dr. Ranga, the podium is yours.
Thank you, Fred, for the introduction, and thank you for the invitation to give this seminar in this webinar. So here I have two objectives for this webinar today. I'm going to briefly summarize the literature on feeding seaweed to dairy cows, and then I discuss some recent research that we did at Iowa State focusing on feeding seaweed to dairy cows. So as many of us already know, agriculture, including both crop and animal agriculture sector, is highly regulated and blamed for most of the times for the emissions to the environment, even though there are so many other sectors contributing to the pollution or the emissions in large scale, unfortunately, but that's the reality. And if we focus on greenhouse gas emissions in the United States, agriculture sector represent only 10% of the greenhouse gas emissions, as you can see in this bar graph. But there are other sectors, transportation in particular, contributing to 29% of the total greenhouse gas emissions. But if you just focus on this 10% and look at the sources of greenhouse gases, the livestock, the direct emissions from livestock production, including enteric fermentation and the manure management, represent about 40% of the total agricultural greenhouse gas emissions. So the enteric fermentation is primarily, the emissions from enteric fermentation is primarily the methane emissions. This is the methane produced during the fermentation of carbohydrates in the digestive tract of animal, and then the methane and nitrous oxide emissions from manure management. If you focus only on the methane emissions, methane is a potential powerful greenhouse gas, having global warming potential 28 times greater than the global warming potential of carbon dioxide. So if you focus only on methane emissions, in US, livestock, including the beef and dairy cattle and swine, contribute to 35% of the total methane emissions of US. And out of that contribution, closely like it's 89%, we can say almost 90% of that emissions comes from cattle, including beef and dairy. And then the majority of that emission originate from enteric fermentations, the fermentation of carbohydrates in the rumen. And if we just focus on the how methane is produced in the rumen for a couple of minutes, or within a minute, I'll try to be fast. So we know that the main function of rumen, or the forestomach of ruminant animals like dairy cows, is to break down carbohydrates and ferment those the product resulting from that digestion, for example, glucose, to produce volatile fatty acids, primarily propionate and acetate, providing 90% of the energy, or the majority of energy, or ATP and glucose in dairy cows. So that's the main function. So these fermentation reactions, particularly the reactions producing acetate, also generate hydrogen. Okay, so the accumulation of hydrogen is not a good thing for the progression of fermentation. Therefore, rumen has to come up with the mechanism to get rid of this hydrogen without being accumulated in the rumen, because if it is accumulated, it will create a negative feedback on the fermentation reactions, the fiber digestion, and then the growth of fibrolytic bacteria, which is essential component of the rumen. So some of this hydrogen is utilized by fermentation reactions themselves. For example, production of propionate utilize some amount of that hydrogen, and also biohydrogenation of unsaturated fatty acids also utilize some a proportion of that hydrogen. But the main mechanisms that removes hydrogen from the rumen is the methane production. Therefore, methane production is not the bad thing, because it has a purpose. It helps the continuation of this very important function, the fermentation of particularly fiber derived component in the rumen, providing energy to the animals. Okay, so this special group of bacteria called archaea perform methane production using this cascade of reactions, and finally releasing methane um, um, as an end product. So each step is catalyzed by specific enzyme. For example, this last step, releasing methane, is catalyzed by methyl coenzyme M reductase, or MCR enzyme. Okay, so now this description, this diagram suggests some mechanisms that we can leverage to mitigate or decrease methane production in the rumen. And one thing is we can decrease the substrate supply for the fermentation. So that means we can, cut down the nutrient intake or the dry matter intake. So that will decrease the methane production, because dry matter intake is the major drive of methane production. But it may not be a sustainable strategy if it is going to sacrifice the production, right? Or unless we improve the efficiency, feed conversion efficiency of animals. Or else we can also enhance other mechanisms utilizing hydrogen in the rumen. For example, enhancing propionate production relative to acetate production, or kind of, inhibit some downstream reactions of this methane production pathway, and thereby inhibit the release of methane. Okay, so there are possible mechanisms. So we have been testing several dietary interventions kind of connected to those mechanisms in the rumen to mitigate methane production of dairy cows. So now in these diagrams, kind of rank their efficacy or the percentage reduction in methane production. For example, we have tried increasing dietary fat content, adding ionophores like monensin, try to improve forage quality, replace fiber with starch, but this ranking says that the feed additive 3-NOP or 3-nitroxypropanol, we just a few weeks ago it got the FDA approval for the commercial use, and also red seaweed has the highest potential to decrease methane production, methane emissions from dairy cows or cattle in general. The 3-NOP inhibits this, the enzymes catalyzing the last step of this cascade of reaction, and thereby inhibit the release of methane. And then the seaweed and the compound in seaweed called bromoform is found to interact with the substrate of that enzyme and then again inhibit the release of methane. Therefore, these two are working at the downstream, very downstream reaction of this methane production pathway in the rumen. So here I have some plot, kind of plot called forest plot from a meta-analysis that we conducted in our lab recently. So this summarizes the literature on the published studies focused on feeding different species of seaweed at different doses on methane production reduction in lactating dairy cows. Overall, two seaweed species, Asparagopsis taxiformis and Asparagopsis armata, both representing the red seaweed group. They have this red pigment in them, so they appear very reddish, feathery seaweed. So these two have shown the most promising result in terms of mitigating methane production. And then the Asparagopsis taxiformis usually grow in subtropical, tropical and subtropical environment, whereas Asparagopsis armata usually grow in warm temperate regions. Some literature indicate that Asparagopsis armata has very high concentration of this active compound bromoform compared to Asparagopsis taxiformis. But again, some other literature indicate that depending on the variety and depending on the environment and the composition of the ocean or seawater, things, those concentration can be quite variable. So Dr. Ermias Kebriab lab at University of California, Davis kind of pioneered in testing red seaweed in dairy cows. They fed Asparagopsis armata harvested from Senior New Zealand, and then they fed that to cows at 2 inclusion rate, low dose or low inclusion rate, that is 0.5% of dry matter, and then the high dose or inclusion rate was 1% of dry matter. However, they observe significant decrease in dry matter intake as they increase the inclusion rate. For the low inclusion rate, they observe 11% reduction, and then high inclusion rate, they observe 38% reduction in dry matter intake. But that 11%, the low dose 11% dry matter intake reduction did not compromise milk yield, but the 38% reduction in dry matter intake decreased the milk yield by 12%. And then the bromoform concentration, which is the active ingredient concentration, um, um, active ingredient concentration increased in both cases when they introduce that product to the diet. However, the elevated concentration is still below the EPA bromoform threshold for beverages, which is 0.7 ppm. And then the interesting part of this study is to see the efficacy of in terms of methane mitigation. So they observe 26% reduction in methane production. Okay, so the methane production, I'm going to use two terms in this presentation related to methane emissions. One is methane production, which is the gram of methane per cow per day. Okay, and then the second term I'm going to use is the methane yield, which is the gram of methane relative to dry matter per kilogram of dry matter intake. Okay, so let's look at the methane production response first in this study. So for the low dose or the 0.5% dry matter inclusion rate, the methane production decreased by 26%, but when they increase when they double the inclusion rate, then the methane production reduction decrease by 67%. Okay, so now we know that this product also decreased the dry matter intake. So, and we also know dry matter intake is, or the substrate supply is the key driver of enteric methane production in the rumen. Therefore, some of those reductions should be confounded in those dry matter intake reduction. So to kind of, to get a refined estimate of methane production by canceling of those confounding effects is to express methane production relative to dry matter intake. When they do that, still the low dose decrease methane production per kilogram of dry matter 20%, and then the methane production per kilogram of dry matter, in other words, methane yield by 43%. It's a good reduction. However, we saw some compromise milk production and dry matter intake associated with those methane emission reduction. The other group of scientists working working on feeding red seaweed to dairy cows is the Alice Ristoff group at Penn State University. So they publish this paper in Journal of Dairy Science, and I think it's the paper this is the paper that Dr. Ristoff received the highly cited paper award last month at ADSA meeting. So this is very highly cited paper. And then in this study, they fed Asparagopsis taxiformis, the other red seaweed species, to dairy cows. Again, 2 doses, but this time they are towards the lower side of the inclusion rate. Their high dose was the low dose of the UC Davis study. So their high dose 0.5%, and then low dose was half of it, 0.25%. They didn't see any effect of the low dose on dry matter intake, milk yield, milk composition, or the methane emissions. But the high dose decreased methane production and yield about 30 to 35%. That's a good reduction, which is similar to what we achieved with 3-NOP. they also experience the reduced dry matter intake, which was also associated with similarly reduced milk yield, but not as high as the reductions that the UC Davis group observed. And but UC Davis group observed a higher bromoform concentration in milk, but the Penn State group did not observe any difference between for any dose at the low high dose. Why we are concerned about bromoform in milk? Because bromoform um, in some experiment, some say like a human focus experiment, have been shown some carcinogenic effect. So that can, I know, high concentration of bromoform therefore can affect the milk safety. Okay, I think I need to, maybe I go back to my previous slide before coming into, but now we see that this the dry matter intake reduction, potentially suggesting, some diet palatability issue, and then associated milk production reduction, something that we need to fix before accepting red seaweed as a sustainable solution to mitigate methane emissions from dairy cows. So that's kind of clear. But the bigger challenge would be the mass scale production of seaweed to feed the total cattle population. For example, 100 million cattle, both dairy and beef, in the US, because all those experiments have utilized wild stock of Asparagopsis taxiformis or Asparagopsis aromata. So the wild stocks are not, I mean, they are barely enough to feed even the cows in experiments. So that means the mass scale production, it can be, again, cultivation in the ocean or offshore cultivation or ground-based. Either way, there should be a way to produce high quality in the sense that the product having that active ingredient at desired concentration, bromoform at desired concentration. So if somebody is going to uh, take that challenge of producing red seaweed in large scale, again, they have to screen many varieties, because we don't know the varieties, the wild varieties would be suitable for example, ground-based tank culture systems, because it's a total different system, right? You need to understand what are the requirements, what kind of water quality, what kind of nutrients you need to add to the media, those culture medias. And so there's a lot of screening of different varieties to find the most suitable one, the suitable in the sense give you a good yield and good concentration of bromoform. So this one company, its name is Blue Ocean Barnes, located in Hawaii, kind of took this challenge, and they worked with scientists. For example, Dr. Jennifer Smith. She's a marine botanist at University of California, San Diego. The picture to the left, you see her in that picture. So this company worked with those kind of scientists who have the experience of grow enost with the seaweed. They screened several varieties and then found this variety, Asparagopsis taxiformis variety called Brominata, to be a really kind of suitable variety for the ground-based tank culture system that give them a good yield with good concentration of bromoform. So recently, the California Department of Food and Agriculture, CDFA, authorized the commercial use of dehydrated brominata. So you see the picture to the right on those trays, you see that the dehydrated brominata for cattle in California. So the recommended or the approved inclusion rate is 0.3% of dry matter. So I'm going to call this approved rate is the standard inclusion rate, or SIR, in my upcoming slide. So now this inclusion rate is based on the literature data and then some data coming from an organic farm utilizing mostly a 100% forage-based diets. Um, but this product needs to be tested, for instance, in TMR diets that we usually fed to dairy cows, and you need more data, in other words, to derive robust conclusion about its efficacy and then also its safety. So that is why the Blue Ocean Band collaborated with my group, my lab at Iowa State, to evaluate their product, the brominata, in lactating dairy cows, and to see how it affect methane emissions, and also that you know what's that also study more about that palatability issue, and also the safety and tolerance of the product in dairy cows. So therefore, one of the questions we want to ask or to answer in this study was to see what is the efficacy of this product at standard inclusion rate when we feed in a TMR diet. So that means we had control treatment, and also we had another treatment representing this SIR. We also want to answer questions like, what if we increase the dose, maybe 1.5 times, 2 times higher than the SIR, whether that is going to adversely affect cows. If they have adverse effect, how long cows take to kind of recover from those adverse effects. So therefore, we introduced two other doses, like 1.5 times of the standard inclusion rate, or the standard dose, or the approved dose in California, and also the 2 times of that SIR to answer those questions. So we use 48 late lactating, that means low producing cows, having average milk production of 25 kilograms per day. Because this is a safety and tolerance type of study, we really don't want to use our best cows, or the high producing cows. So because this product has not been tested in a proper dairy cow setting, with lactating cow setting before. And so we had to, it's the complete randomized design. So we have 12 cows per treatment. And then we use 4 green feed units to measure methane, a green feed unit to methane, to measure methane emissions. So we started our study with a 10-day training period. So this is to train cows to Kalen gates, because we use Kalen gates to measure individual feed intake. And then we utilize these 10 days to train cows for the green feed system, because cows have to visit at least 1 or twice per day to get a good methane emission measurement over the time. Okay, so that's the training period. And then following that training period, then we had the baseline measurement period, where we measured their production, methane emission, and some health traits that we are going to measure again after introducing the product. And then we had inclusion rate ramping up period. It's more like an adaptation period. So we introduce their expected inclusion rates gradually. That means 28 steps, like, each day we increase the inclusion rate by certain percentage. By 28th day, we reach their desired inclusion rate. For instance, 0.3% of dry matter for SIR, 0.45% for SIR 1.5, and 0.6% of dry matter for SIR 2 treatments. And then we continue with those inclusion rates for next 63 days. That's where we measure the responses to treatment. So those are the responses going to be in our statistical models analyzing treatment effects. So um, so the brominata was introduced to the diet as a pellet. So this pellet was made of 20% brominata, and then the 20% distillery solubles, and about 60% of the wheat middling. So this pellet was made in our new feed mill, Kent feed mill. This is one of the first product that they made for research at Iowa State University. So again, our main responses variables, we measure daily dry matter intake using Kellen gates, the milk yield, and then the methane emissions using the green feed system. If you're familiar with how green feed system works, you know that green feed system use a type of pellet to entice animal to the machine and give some gas measurements in their breath, right? So that means in addition to the dry matter they consume from their bin, they also consume some dry matter from that comes with those bait pellets in the green feed machine. I just want to emphasize that. And then we weekly recorded some health scores. We recorded body condition score, the locomotion score, the breathing scores, and then the fecal consistency, some weekly health, which indicate their safety and tolerance. And then every other week, we collected milk samples for general milk composition analysis, as well as the milk bromoform analysis. And then we collected blood samples in the midpoint and the final point of the study to analyze for the blood chemistry and the hematology, again, to describe the health and safety effects. And then we also collected feces on 2 consecutive days at 5, 13, and 25 hours on each day to determine the total tract digestibility of nutrients. And then after 63 days, we stop feeding the product, but we still follow up those cows for another 14 days to understand, to answer the question, how much time the cows require to recover from any adverse effect of the product, for instance, on dry matter intake or the milk production or composition. Now I'm going to present you some results. Here I have the dry matter intake. So now again, dry matter intake has 3 components. One is the dry matter coming from basal TMR diet, representing 96% of the total dry matter intake, and some like 4% of dry matter coming from the green feed pellet, and very little, like 0.4% dry matter from that pellet carrying the bromoform. So now when we increase the brominata inclusion rate, the dry matter intake decrease linearly significantly. So the standard inclusion rate had a 15% decrease in dry matter intake, and then 1.5 to add further decrease in dry matter intake. And then in methane emissions also linearly decrease. Methane production decreased by 46% for the standard inclusion rate, and then the reduction was further intensified up to 36% when we increased the inclusion rate to 2% of dry matter. But again, you know that the part of this reduction should be confounded in the dry matter intake reduction. So that is why we need to look at the methane yield, which is adjusted for the dry matter intake change. When we adjust for the dry matter intake change, still the standard inclusion rate was associated with 35% reduction in methane yield, and then the higher doses had 50 to 60% reduction in methane yield. Now, the milk yield and milk component yield, and then the composition-related traits. So the milk yield, despite that 15% reduction in dry matter intake, the SIR did not experience any milk yield reduction, which is similar to what the UC Davis group observed. But when you increase the the inclusion rate further, particularly to 2 times of the standard rate, so it compromised milk yield by 30%. And then fat percentage had kind of linear reduction, particularly at very high, the 2 times of the SIR, you begin to see a significant reduction in fat percentage, but the SIR, there's no significant difference between the fat percentage of control and the SIR. And then the bromoform concentration stay the same. We know there's no much, there's no change in bromoform concentration in milk, which agrees with the Penn State data with that feeding taxiformis did not change the milk bromoform concentration. This is maybe because most of the bromoform you feed to dairy cows are metabolized in the rumen, so so therefore the amount of bromoform absorbed into the blood would be minor. And then the body weight, there's no significant difference in body weight change during that whole 63-day period in this experiment. And then if you look at the treatment effects on total tract nutrient digestibility, there's mostly the cubic relationship between the seaweed dose and all the nutrient digestibility, that the maximum digestibility was achieved with standard inclusion rate, while the minimum was achieved with that 1.5 times the SIR. But if you just compare the values to the control treatment, the SI has there's a tendency for standard inclusion rate to increase the dry matter, organic matter, and NDF digestibility. So that may explain, at least partly explain how cow maintain the constant milk production despite the dry matter intake decrease, because their digestibility, they try to compensate for the nutrient supply by improving the nutrient digestibility here. And the health scores, we saw that linear increase in locomotion score as we increase the higher locomotion score reflected the locomotion issues. That means lower scores reflect the better the locomotion traits. However, it's just still, even though now you see that in control 1.23, but the highest dose is 1.58, still below 2. So um, even though it's a significant linear relationship, but the in terms of the health or the biological interpretation, I don't see it's anything alarming here. The body condition score again linearly decrease with increasing inclusion rate, but again, all the values are close to 4 overall. So not the huge mobilization. And then the scores were quite similar among treatments. And then I know this is a very busy slide. Um, but the take-home message is that all the blood chemistry parameters were within the usual, the normal physiological range or the reference range that we got from our veterinary diagnostic lab, even though some treatment effect, for example, actually some treatment effects are, it's favorable. For example, when you increase the brominata inclusion rate, the alanine transaminase concentration, which is a indication of liver damage, decrease. So that's good thing. But anyway, just all the values are within the reference range. The same thing with the hematology, all the values are within the reference range. Okay, now some data, about the recovery, uh, uh, the recovery period. So this is the measurement that we obtained the second week of or the 14 days after we withdraw the product from the diet. Now you can see that dry matter intake has, bump up to their normal intake level. There's no difference among treatment when you measure the dry matter intake 2 weeks after you stop feeding the product. The milk yield, uh, rate had come back to their usual level, but remember they are very late lactation. You cannot expect them to produce too much, but they are around 20 kilograms of kilograms per day milk yield, but no treatment difference. Fat content, no treatment difference. And then the methane production, there are some numerical differences, but there's no statistically significant. So that means within 2 weeks, if those adverse effects on dry matter intake, milk yield, and milk composition um, um, could be corrected in case the cow accidentally consume high dose of the brominata, if you think about that kind of a scenario in a farm. In summary, our results indicated that brominata at standard rate, that means 3% of dry matter decrease methane emissions by 35 to 45%. That's a decent emission reduction without affecting milk production or health, despite the 15% diameter decrease. So we could achieve a further reduction, up to 60 to 75% emission reduction by increasing SIR above or the inclusion rate above the SIR, but however, it badly compromised dry matter intake and milk yield and composition. So then overall, the brominata feeding at SIR level in the TMR diet has a potential to decrease the methane emission without affecting the milk production or the health. But again, I want to emphasize that these data coming from low producing, late lactating cows, but the similar data, the more data, particularly from high producing cows, will help determine the true potential of brominata as a sustainable methane mitigation strategy for dairy cows. So that's the summary. And then I want to acknowledge my grad and undergraduate student assistant received from ISU Dairy Farm and the funding received from Blue Ocean Bands. And thank you very much.
Thank you very much, Dr. Ranga. We do have a couple of questions that have come in. Larry, do you want to go ahead and ask the first question?
Yeah, just has any economics been done on any of this, like the cost of the seaweed, or is there any calculators that could be created or should be created just to look at dry matter increase or the decrease in the dry matter, the decrease in the milk production, and just what economic benefit or what environmental benefits can you put any values on
Yeah.
the emission part of it?
Not that I aware of, but I think that's a really a good point that actually we can do that, not like in a perfect manner, at least to some extent. So based on the the feed prices and the milk prices, taking our ISU dairy farm as an example. So yeah, that's a good point.
And then Jen had a question.
Yeah, maybe Ranga, you had answered it already. Towards the end, you mentioned that these cows were late lactation. So I was just curious on any effects on reproduction, but maybe these were already pregnant cows.
Yeah. The one of the cows in high dose had an abortion, but she had the history of having abortion. I don't think that is the impact of the product. Uh, but however, we are going to follow up those cows, in a week or something. I'm going to shoot an email to Mary and ask, any recent record of, reproduction related records, and then I think we can look into those kind of effects, based on whatever the data we have at the farm. Yeah.
We've got a couple other questions here. First one's kind of interesting. Feeding seaweed and 3-NOP, would that complement and compound the effects, or hasn't there been any determination on that?
Oh, that's a really good question. And actually, uh, there are some research funding that emphasize the combined, investigations focused on combined effects of strategies. So I think this come under that maybe focus, but I haven't seen any literature doing that. But what I hypothesize, looking at, if you look at the mechanism of action, the where they kind of, target in that methane production reaction cascade, they're kind of working around the same reaction step. Can be synergistic, or it can be complementary. So I'm not sure, but I feel like it is not going to be additive. Yeah.
Okay, very good. You mentioned a couple of times that the seaweed requires some processing. Is this something that you dehydrate it and pelletize it, or what's involved in that process?
Yes. So, yeah, dehydration, and then the pelletization. The pelletizing is two main process they underwent we feed before we feed the pellet to cows. So now these two processes can remove some of the bromoform, the active ingredient from brominata, because the bromoform is easily volatilized compound. So the Blue Ocean Barn has developed and sort of the model or the algorithm to account for those kind of events, the effects of those kind of events on the bromoform concentration. So I think that's for the first time, I think somebody's developing, some way of estimating the final bromoform concentration, because sometimes it's the people report the bromoform concentration um, in the harvested products without accounting for the post-harvest losses. So I think I don't have that number in hand, but in 2 weeks we are going to have a Zoom call to discuss that algorithm, and then because I need that for the publication. Yeah.
Okay. You mentioned that this research was done on late lactation cows. Can you make any uh, educated guesses? Is this a product that if we started with heifers and as they came into transition pen, got them started, will we see some increased benefit, or is the long-term effect not necessarily explained yet?
Right now, in production performance, the animal performance wise, the challenges is this, the issues are the reduced dry matter intake, which explain the reduced milk production. But the feeding this to heifers, for instance, would, we don't have to worry that much about that dry matter intake reduction and it connection with milk production. I think this, even could be even more sustainable strategy to cut down methane emissions from heifers than lactating dairy cows. But I don't know, but we need because there's a study that currently happening at Washington State University. They are that is focused on looking at the effect of the brominata on whole lactation. So that data from that experiment explain better how that is going to affect the lactating cows. But based on this late lactating cow data that we have, I hypothesize even more sustainable impact on heifers.
Okay. If folks have questions, we'd invite them to go ahead and type them into the chat box. That way we can make sure and get as many questions answered as possible. We have another question. Broniform, is it natural in the ocean, or is it an effect of pollution? Or where is it coming from in the seaweed?
Seaweed has a special kind of a glands that produce the bromoform. And again, as I mentioned, bromoform is the volatile compounds. So, the seaweed can release bromoform to the environment. And then, some studies have shown that the bromoform can kind of damage ozone layer. So this bromoform volatilize from any source could not be that good to the environment.
Okay. Does this specific seaweed require salt water, or is this a potential thing that I can raise in my pond?
So I'm not a marine botanist, so I don't know what culture conditions, these different species require, but I know it is a tedious process to screen different varieties or strains and finding out the best variety that's suitable for your purpose. In the sense the culture environment you have, say your pond, and also what you expect from it. In industry, the seaweed in culture industry expect a good bromoform concentration in it.
Okay. Final question that I have is, you talked about inclusion rates.
Mhm.
Is that for maximum reduction or maximum palatability, or how is that determined?
Yeah, this 3%, I'm not sure about the total background associated with the determination of that 3%, but I understood was they look at the literature as well as the data they have um, um, in California to determine 0.3%. But if I do, such a determination, I'm sure they did the same way. It's not the maximum, it's the optimum. So now you see that our 0.2 resulted in maximum emission reduction, but it compromised milk yield, dry matter intake, and milk yield, and some milk composition traits. So therefore, you need to think about holistic, way to more like to determine, how you make most of it, sustainable manner, not just the methane emission reduction, but also the other traits that is important or represent in the sustainability.
Very good. Larry, Jen, did you have any additional questions that you're seeing?
Not that I'm seeing. Good here.
Okay. If not, again, I'd like to thank Dr. Ranga for the presentation. A lot of information to absorb. I appreciate the way you made it so even I could understand most of it. It's always appreciated. I'd like to remind everybody that this webinar will be archived at the Iowa State website. And in addition, click on the short survey. It's only 4 questions, kind of help us get a handle on what is important and what we need to change. With that, I'd like to mention that we will be having a biosecurity guidance for dairy operations webinar on July 25th. It'll be hosted with the I-29 Moo University. And then in August, we will have another webinar. We're going to talk about how we manage colostrum during the months when we tend not to get as much or as good a quality colostrum. So until August, we thank everybody for attending today and look forward to seeing you next month.